Okay, so sorry for the slight delay before restarting. So our next speaker is Maria Leptin from EMBL, and she will be telling us about tissue folding, cell shape changes in folding epithelium, genetically and mechanically determined boundaries. So thank you, Maria, for coming.
Thank you for the invitation, Thomas, and it's great to be here. And it's nice, well, what I'm going to be talking about is all completely new. I'm talking about it for the first time today, and I only decided that this really worked well for this meeting very recently. So it's a little bit messy, and my preparations have been marred by failure to concentrate sufficiently in the background of having to watch Brexit news. But I will be talking about a topic that's been sort of on and off my agenda for most of my independent career, and that is epithelial folding. And the case in point is the invagination of the mesoderm in the early Drosophila embryo. The Drosophila embryo is fantastic in many ways, quite apart from simply being beautiful. Partly because we know so much about the early genetic program. For instance, we know all the transcription factors and their expression domains that determine the earliest morphogenetic events. They're shown here, so you see a whole lot of transcription factors shown in different colors and in different domains that each have a responsibility for a particular example of epithelial folding. And what they do is control and direct these cases of epithelial morphogenesis. So they make the head fold, they make dorsal fold, they make the invagination of the mesoderm on the ventral side of the embryo, and they make the invagination of the endoderm at the posterior end of the embryo. And you see underneath that we understand many of the components of the subcellular components that are under the control of these transcription factors. We also have a non-folding event, but a tissue rearrangement event, of which we also understand the genes that direct them. So it's really fantastic. We really know the upper control level, and we know much of the cell biology of the downstream level. But there remain a lot of unanswered questions for which the system is great, precisely because we do know so much. So the one I'll be talking about is what happens in that huge red area, which is the area in which two transcription factors are expressed, twist and snail. And it is their responsibility to lead to the not only the early morphogenesis, but also the later differentiation of the mesoderm, the future mesoderm. And at the top here are two very old images of a Drosophila embryo before and after gastrulation, before morphogenesis has started, any morphogenesis has started, and at the point when early morphogenesis has created the germ layer. So it's brought the brown cells from the outside to the inside. And we know that this is mediated, which one is the pointer? This? Okay, no, here. Okay, right. So we know that this is mediated, as shown here in cross sections, through the middle of the embryo by cell shape changes on the ventral side in the area that expresses both transcription factors, twist and snail. And these cells undergo a series of stereotypic cell shape changes that change the shape of the epithelium and then bring the cells inside. We also know, Danielle, what a pleasure. We also know and have determined, that's two labs, Eric Wieschaus's lab and my lab, have determined the, first of all, this is mediated by transcription factor twist. Second, we've determined the downstream targets of this transcription factor, which are shown here. Two of them are permissive, the others are instructive. And these genes together instruct the transition of the cells of the mesoderm to undergo this transition from box-shaped to wedge-shaped. And the way they do this is by acting on a series of proteins, a whole set of proteins that exist within the cells, So the ones that are shown here that are turned on specifically in those ventral cells are circled here in blue, and they act upon a whole set of morphogenetic molecules that are present, that pre-exist in the cells. So folded gastrulation fog is a secreted ligand. It's secreted by these cells and then binds back to a receptor mist that's also expressed here. It activates a G-protein coupled receptor, which then activates a downstream cascade via ROGEF. It is counteracted by a kinase here, a GPR receptor kinase 2, which I'll come to later. Another downstream target is a transmembrane molecule called T48, which acts as an anchor for ROGEF. So together, this module here activates the Rho signaling pathway, which in turn leads to the activation of apical myosin. A separate pathway acts on adherence junctions and makes sure they are localized very tightly to the most apical site. You'll notice the cell is upside down now, so sometimes ventral is up, sometimes ventral is down. Unfortunately, two conventions have developed. Okay, so the other pathway acts on the junctions. That pathway ensures, like I said, that the junctions, which initially are subapical, you see here a cross-section only of the ventral part of the embryo, which is stained for junctions and for myosin. So one pathway ensures that the initially subapical junctions are very tightly apically located, where they act as an anchor for the actin cytoskeleton, which will contract. And indeed, the other pathway leads to the assembly of this actomyosin meshwork at the outer apical side of cells. And this is an image from Adam Martin when he was still in Agrichas's lab, and which shows that this contractile actomyosin meshwork leads to the constriction of the apical side of these cells, which creates this indentation. So we know that the forces that make this furrow are generated within these cells. There are no forces that arise from outside. This is really an autonomous activity of these cells. But we've really understood only these cells. While these may not be instructive, they at the very least have to be compliant. They have to be permissive. And so we wanted to understand how the rest of the embryo interacts with this morphogenetic event to allow it to happen. And when Matteo Rauzzi, who at the time was a grad student in Thomas' lab, joined the lab, that's what he set himself as a goal, to understand what the entire embryo does in order to accommodate this invaginating tissue. And he he first set up his own microscopes and did a lot of work on that, but in the end ended up collaborating with Lars Hufnagel to use a multi-view selective plane illumination microscopy to observe the embryo at very high temporal and spatial resolution to be able to follow cell shapes. And here is an example of the type of, have I really talked for 9 minutes? Shit. Okay, I'll skip this. This is a data set. What we did from this data set is extract only the surface of the embryo, which we then present in a cartographic projection. So this is an embryo that's basically unrolled, and you'll be able to watch the ventral furrow form in the middle of the embryo. So you see here, this is where the ventral side of the embryo, here's the cross-section through the middle. And you'll see that these cells constrict and the rest of the cells flow in. You'll see the germ cells doing their thing with the posterior midgut to go inside the embryo. An easier way to look at this in one image is a chymograph, where we just take one plane and then follow this section throughout time. And what you see then is that there are two phases. Initially, nothing happens. Then these ventral cells go inside while nothing much else happened. And in the second phase, after the ventral cells have moved in, two cohorts of lateral sides flow towards the middle while the dorsal cells stretch. So there are several cell populations that act during various phases. The central cells go inside, disappear from the surface, and then later the lateral cells shift. And we've also measured their properties. They differ in their mechanical properties. Now, if these cells dive inside and these don't budge, the embryo would pull itself apart. But of course, it doesn't. And the reason for that is these green cells. And what they do is, while these cells constrict and these just sit still, these actually respond by stretching. And you can see this very clearly in the cross-section. And I'll concentrate on those cells for the rest of my talk. They are what graduate students who are at Beta concentrated on. And they're really interesting. First of all, they're interesting for biological reasons. They're very ubiquitous. They exist throughout the animal kingdom. We call them hinge cells because they act as a hinge between this and that. And for example, they exist here in Volvox, where you have these kinds of bottle cells, and you see that the lateral cells do exactly the same thing. They stretch around to allow this constriction to take away stuff from the surface. They're extremely hard to image because not only do they move very fast, they also move over large distances. And this is a central cell, which is easy to follow and has been done by others. These are the lateral cells. They're very hard to image and to segment because they move over a very short time, over a very long distance, more than 100 microns, and you have to resolve this at less than 20-second intervals to great depth. Anyway, Sorab managed to do that, but it still poses large difficulties. Anyway, so these stretching cells, this is again Matteo's movies now filled in for cell size, for just apical surface area. And what you'll see is that the cells, and I'll run it again. Can I run it again? No, I won't run it again. So by the end of when the furrow has begun to invaginate, you see that these cells have stretched very much, but then there's a sharp border to the next row of cells which doesn't stretch. It does run again. If we look at this at a still and from a different movie that is more easily segmentable, we see this border. This is slightly earlier, so it's not so pronounced. But we also see a border here between these stretching cells and the cells that have constricted in the middle. The ones that are just slightly green here or even orangey, they will constrict in a couple of seconds' time. So this is a bit surprising if we look at the gene expression patterns about which we know in the animal at this time. So I'm showing you this again. So all of these cells will eventually become mesoderm. These will become ectoderm. And we know genes that are expressed in these. The most explicit case is snail, which really defines this border. We know genes that are expressed in these cells that are neither fish nor flesh, so they're neither ectoderm, they're called mesectoderm. They express a gene called single-minded, which is relevant in a moment, but which has no functional role at this time. You get rid of it, nothing changes. But we have these totally different cell behaviors here, massive stretching and extreme constriction, and we have no genes that distinguish between these two cell types. The only thing we have are ones that are expressed in the gradient. So twist itself is expressed in the gradient, and twist's downstream genes that I introduced earlier are expressed in gradients, with a peak in the middle. But there's no border here. In fact, I quickly want to turn to fog. This is beautiful work that was begun in the Levine lab that shows the fog expression over time. So it ends up in a gradient, which you don't see here, but the intensity gradient actually occurs by being built up by sequential accumulation of cells expressing fog over time, which reminded me of the first ever 4D movie that was taken of Drosophila gastrulation in 1991, which showed a similar stochastic behavior. Now, in 1991, there was no GFP around. So what did we do at the time? We injected fluorescent dextran in the paravitelin space, which marks cell outlines, and rhodamin-labeled histones into the inside, which becomes incorporated into the nuclei. And what we discovered is that the apical constriction of cells was accompanied by nuclear movement, and so we could therefore use the nuclei as proxy for cell shape change. And what we discovered is that if we look the disappearance of nuclei from the surface of the embryo, then we found that that was a stochastic event with a higher likelihood to start in the middle. But even here's one way out, and when one's inside haven't gone down. So there is a stochastic movement of nuclei, of cell shape change in this area, which probably mirrors the accumulation of folded gastrulation and other molecules in the cells. This may be relevant for the discussion later. Okay, so we wanted to look a little more closely at the shape changes in these hinge cells. And what Sorab did is he just looked at 3 rows of cells, the outside one, which is this mesectoderm cell, which, by the way, has a subapical junction on one side and an absolutely apical junction on the other. I'll show you again when we return to a larger scale image. And so he just looked at these cells and asked, over time, how do they change their shape? And you see that the outermost one very gradually and gently changes. The next row here also hasn't done much. And the inner one changes a lot more. And we can quantify that, the end point. At the end point, we have the cells in the middle that have constricted, the ones outside that haven't changed. And then we have this gradient of cell size going from lateral to central. Now, that's counterintuitive. And just to show this, we did a little cell movie. If the force comes from the middle and pulls on the outer cells, then what you would predict if you were looking exclusively at elastic activities, well, what you would predict is what we just sort of exercised through with a little model where we have different cell populations, and then we impose different contractility. In fact, we impose a bell curve of contractility that increases over time. And what you then see is the following, namely that you get a stable minimal energy situation where you have strong constrictions in the middle, and then increasingly larger cells and a stretch here. We do not have these cells larger than these. Not surprisingly, anybody could have predicted that. So we thought, what do we have to do to change that? Just in terms of toy models. Still, even if you flatten the gradient outside, then you end up again with a bell curve of sizes here and equal sizes out here. And what we would have to do to get what we observe is to inverse the gradient. And so then you could, in theory, achieve what we see in vivo. But there is no evidence there's no data at all that suggests that this exists. So it has to be something else. So this really raises a bunch of questions. So why is there a sharp boundary over an underlying smooth gradient? We know nothing, and I tell you, the Drosophila early embryo has been looked at for gene expression patterns ad nauseam. I think we really have a full image of everything that is expressed. There's nothing that has a boundary within the mesoderm. So why a sharp boundary over an underlying smooth gradient? Why do the shapes not reflect the gradient? Why do they inverse? And what's active and what's processed in this? So we explored a little bit these virtual and potentially real boundaries. And ask, is the genetic boundary at the side? And I have to watch my time. Okay. A real boundary or not? Are we imagining something? And what determines, really, that's the million-dollar question, what determines the border within the mesoderm? I won't be able to answer that. I won't show you data on this one, on the genetic boundary. I'll just report some facts. So why does the ectoderm not stretch? It could be because it's just too far away because there's viscosity in the system and it's too far away from the source of the force. That is unlikely because in a certain type of mutant embryo where the whole embryo thinks it's mesoderm, but you still get a furrow forming on the ventral side, then the lateral cells stretch. So the force is sufficient to reach the lateral side. A different example is a molecule that dampens the G protein signaling that leads to Rho activation, GPRK2. If you take that way, you get a wider domain. The entire domain tries to constrict, and you actually get a big domain. But in that case, there's still almost no stretch in the ectoderm. So we think this together suggests, and there's more work to be done, and there's more data as well, that is really a genetic boundary that distinguishes the ectodermal from the mesodermal cells. And we'll see a little more on that. Okay, so what about the border within the mesoderm? What I want to propose, we don't know, but what I want to propose and show you some supporting data is that it's a hypothesis that it's a kind of a tug of war. So we think that perhaps all cells have the capacity to constrict, to do the same thing. They're equal, they have the same program, just at different levels. But some win because they're stronger and thereby inhibit the weaker ones from winning. So it's a competition. Now that makes predictions. For example, if we were to inhibit the strong cells, then the weak ones ought to be able to show their capacity and constrict. And similarly, you could give the weak ones a head start and ask, what does that do to the strong ones? And to do that, we used optogenetics. I think I'd better not explain this. It's a system that was developed in HeLa cells, but was adapted for Drosophila by the Dorenzis lab, and it exploits light-sensitive dimerization between two components, where one is fused to GFP in the membrane, and the other contains a phosphatase that will nibble off the phosphates of PIP2 and thereby disallow the cortical actomyosin meshwork to attach to the membrane. So if you use that, so here's the phosphatase attached to M. cherry. It's homogeneously distributed. These black spots are the nuclei, so it's not in the nuclei, but it's certainly not membrane associated. If you illuminate with blue light and lead to the dimerization of these two components, here's the one component, this is the membrane-anchored component, you will see that here now the phosphatase associates with the membrane, and what you see is that this embryo, which should make a furrow, doesn't. And if you look at the end of the process and look at a cross-section, you see that it's not completely inhibited. You see some constricted cells here. We'll get back to that. But you see that cells right near these mesectodermal cells are certainly able to constrict, which they otherwise never do. And if you look at a ventral view and mark these, redden the mesectodermal cells and these lateral cells, mesodermal cells, they're all small. Even somewhere there's one. Here's a mesectodermal cell that's as close to the mesoderm. We'll get to that. So this is crucially dependent on knowing what is mesectoderm and what isn't, and we can in fact determine that by using a very neat system, again from the Levine lab, using this beautiful RNA visualization method, where we can see nascent single-minded transcript in these. So we know for sure these are the mesectoderm cells, and the blue ones therefore for sure are mesoderm cells, and they constrict, even in situations like these where the mesectodermal cell is as close to the mesoderm as are others. So clearly, there's a genetic distinction at this boundary by which lateral mesodermal cells are able to constrict, that was premise one, but also that the ability to constrict is limited to the mesoderm and the ectodermal or mesectodermal cells cannot. What about the other way around, giving the weak ones a head start? Here we used a similar, again, implemented by the Dorenzis lab. We used the same system, but this time we don't bring a phosphatase to the membrane, but we bring ROGEF to the membrane, which is what triggers myosin activation. And what Sohrab did here is take an embryo, and in half of it, he illuminated. So he activated this cascade in the lateral cells, leaving the central cells alone. Now, this I'm going to have to manipulate a little bit. Let's run it first, but then let's stop it. So you see the anterior and posterior behave differently. Let me just walk you through it slowly. So you see the anterior has now made a broad furrow. The posterior is trying. They're clearly, the middle ones clearly are trying to do it, to do their job, but they're prevented. So again, you see a sort of salt and pepper of large and small cells, and the posterior ones clearly aren't doing their job properly. And if you look at cross-sections, at the end of the process, you see proper invagination here, beginning invagination here, and here at the back, you see two small lateral areas of constriction. So the central cells actually depend on the lateral cells not to constrict to do their job. So I think this shows really that it is a tug of war. And what we think is happening is that you have a slight unevenness in the actomyosin distribution. We can illustrate that. And then once this contracts, it begins to increase the surface of that one, thereby lowering local actomyosin concentration and leading to this difference. So we have a genetically determined border here and a border here determined by cell-cell interactions. And this fits with models that we've shown. So this is an in vitro in silico model of actin side meshworks, where we find that if you take actin fibers and titrate in crosslinkers or motors, if you shift from a certain concentration of, say, this one, from motor to motor versus crosslinker, if you lower them, you lose contractility. Yeah, 1 minute. Is the gradient necessary? Just last quick point. We think it is. Here is what we see if we downregulate contractility in the movie I just showed you. We get salt and pepper. We don't get nothing. We get salt and pepper. Well, that's not so interesting, so they don't constrict, they don't invaginate properly. But the interesting thing is, if you increase folded gastrulation signaling, and we haven't yet segmented it, it's too lousy, this movie, but you see the same thing. You don't just get a whole thing that invaginates. You get tug of war within the region. And so it seems as if a gradient might be necessary to allow sequential constriction, much in the way that Ed talked about the zippering this morning. Okay, that's where we are, and I really look forward to input from you. Again, the people who did it, Matteo started the imaging, Alessandro did other stuff that's not here, Julia did all the modeling, Sorab did the work on these cells, and we've had help from lots of other peoples and, of course, our funders. Thank you.